What is pharmaceutical analysis? A. The study of manufacturing pharmaceutical drugs. B. The study of analyzing pharmaceutical drugs for purity, potency, and quality. C. The study of prescribing pharmaceutical drugs to patients. D. The study of marketing pharmaceutical drugs to consumers. The correct answer is B. Pharmaceutical analysis is the study of analyzing pharmaceutical drugs for purity, potency, and quality. Which of the following is not a part of the scope of pharmaceutical analysis? A. Assay of active pharmaceutical ingredients. B. Stability testing of pharmaceutical products. C. Clinical trials on drug efficacy. D. Quality control of pharmaceutical formulations. The correct answer is C. Clinical trials on drug efficacy are not typically a part of pharmaceutical analysis. Which analytical technique is commonly used in pharmaceutical analysis for the identification of compounds based on mass-to-charge ratio? A. High-performance liquid chromatography. B. Gas chromatography. C. Mass spectrometry. D. Infrared spectroscopy. The correct answer is C. Mass spectrometry, (EMS), is commonly used in pharmaceutical analysis for the identification of compounds based on mass-to-charge ratio. What is the purpose of dissolution testing in pharmaceutical analysis? A. To determine the melting point of a drug. B. To assess the release rate of the active ingredient from a dosage form. C. To identify impurities in a drug sample. D. To measure the viscosity of a pharmaceutical formulation. The correct answer is B. Dissolution testing in pharmaceutical analysis is used to assess the release rate of the active ingredient from a dosage form. Which technique is commonly used to separate and quantify different components in a pharmaceutical sample based on their interactions with a stationary phase and a mobile phase? A. Spectrophotometry. B. Nuclear magnetic resonance, NMR, spectroscopy. C. High-performance liquid chromatography, HPLC. D. Thin layer chromatography, TLC. The correct answer is C. High-performance liquid chromatography HPLC is commonly used to separate and quantify different components in a pharmaceutical sample based on their interactions with a stationary phase and a mobile phase. Which is a non-destructive analytical technique often used in pharmaceutical analysis to identify functional groups present in a drug molecule? A. Nuclear magnetic resonance, NMR, spectroscopy. B. Mass spectrometry, EMS. C. Gas chromatography, GC. D. O-visible spectroscopy. The correct answer is A. Nuclear magnetic resonance NMR spectroscopy is a non-destructive analytical technique often used in pharmaceutical analysis to identify functional groups in drug molecules. What is the primary goal of validation in pharmaceutical analysis? A. To determine the efficacy of a drug in clinical trials. B. To ensure that analytical methods are accurate, reliable, and reproducible. C. To market a new pharmaceutical product successfully. D. To monitor the adverse effects of a drug post-marketing. The correct answer is B. The primary goal of validation in pharmaceutical analysis is to ensure that analytical methods are accurate, reliable, and reproducible. Which technique is commonly used to determine the purity of a pharmaceutical compound by measuring the amount of UV or visible light absorbed? A. Gas chromatography, GC. B. High-performance liquid chromatography, HPLC. C. O-visible spectroscopy. D. Infrared spectroscopy, A. The correct answer is C. O. Visible spectroscopy is commonly used to determine the purity of a pharmaceutical compound by measuring the amount of O or visible light absorbed. What does TLC stand for in the context of pharmaceutical analysis? A. Total limit of concentration. B. Thin layer chromatography. C. Therapeutic lethal concentration. D. Toxicity limitation calculation. The correct answer is B. TLC stands for thin layer chromatography. It is a technique commonly used in pharmaceutical analysis for separation and identification of compounds. Which analytical technique is commonly used in pharmaceutical analysis to study the chemical structure of a drug by measuring its absorption and transmission of infrared light? A. High-performance liquid chromatography, HPLC. B. Gas chromatography, GC. C. Nuclear magnetic resonance, NMR, spectroscopy. D. Infrared spectroscopy, A. The correct answer is D. Infrared spectroscopy, A, is commonly used in pharmaceutical analysis to study the chemical structure of a drug by measuring its absorption and transmission of infrared light. Which technique is used to determine the concentration of an analyte in a pharmaceutical sample based on its ability to conduct electricity? A. HPLC. B. FTIR. C. ICP-MS. D. Potentiometry. The correct answer is D. Potentiometry measures the voltage or current of a chemical system based on the concentration of ions present, making it useful for quantifying analytes in pharmaceutical samples. What is the main difference between qualitative and quantitative analysis in pharmaceutical analysis? A. Qualitative analysis measures the concentration of analytes, while quantitative analysis identifies the presence or absence of specific compounds. B. Qualitative analysis determines the amount of impurities in a sample, while quantitative analysis focuses on identifying unknown compounds. C. Qualitative analysis identifies the nature of compounds present, while quantitative analysis measures the amount of specific compounds in a sample. D. Qualitative analysis measures the property of the sample, while quantitative analysis determines the physical form of the substance. The correct answer is C. Qualitative analysis aims to identify the nature of compounds present in a sample. Quantitative analysis focuses on measuring the amount of specific compounds in the sample. Which analytical technique in pharmaceutical analysis involves measuring the scattering of light by particles in a sample to determine particle size distribution? A. NMR spectroscopy. B. DLSC, GCMSD, XRD. The correct answer is B. Dynamic light scattering, DLS, is commonly used in pharmaceutical analysis to measure particle size distribution by analyzing the scattering of light by particles in a sample. What is the purpose of stability testing in pharmaceutical analysis? A. To determine the chemical composition of a pharmaceutical sample. B. To assess the effects of temperature, humidity, and light on the quality of pharmaceutical products over time. C. To separate and quantify different components in a sample. D. To measure the absorption or transmission of infrared light by a compound. The correct answer is B. Stability testing in pharmaceutical analysis is conducted to evaluate the effects of environmental conditions like temperature, humidity, and light on the quality and stability of pharmaceutical products over time. Which analytical technique is commonly used in pharmaceutical analysis to determine the molecular weight of a compound by measuring the separation of ions based on their mass-to-charge ratio? A. NMR spectroscopy. B. LCMS. C. DSC. D. Same. The correct answer is B. Liquid chromatography mass spectrometry LCMs is a technique used in pharmaceutical analysis to determine molecular weight by separating icons based on their mass-to-charge ratio. What is the main focus of method development in pharmaceutical analysis? A. To identify the impurities present in a sample. B. To establish new analytical techniques for pharmaceutical testing. C. To optimize and validate analytical methods for specific pharmaceutical compounds. D. To perform stability testing on pharmaceutical products. The correct answer is C. Method development in pharmaceutical analysis involves optimizing and validating analytical methods tailored to specific pharmaceutical compounds for accurate analysis and reliable results. In the context of pharmaceutical analysis, what does ICP-M stand for? A. Inductively coupled plasma mass spectrometry. B. Ion exchange chromatography particle size. C. Infrared coupled preparation mass spectrometry. D. Isolation concentration process molecular structure. The correct answer is A. ICP-M stands for inductively coupled plasma mass spectrometry. It is a powerful analytical technique used in pharmaceutical analysis for elemental analysis and quantification. Which type of validation in pharmaceutical analysis ensures that an analytical method consistently produces accurate and reliable results within specified limits? A. Method transfer validation. B. Internal standard validation. C. System suitability validation. D. Method validation. The correct answer is D. Method validation in pharmaceutical analysis is focused on ensuring that an analytical method provides accurate and reliable results within predefined limits consistently. Which factor is critical when selecting an analytical technique for pharmaceutical analysis? A. Speed of analysis. B. Cost effectiveness. C. Sensitivity and selectivity. D. Ease of sample preparation. The correct answer is C. When choosing an analytical technique for pharmaceutical analysis, factors like sensitivity and selectivity are crucial as they determine the method's ability to accurately detect and quantify target compounds in complex samples. What is the primary purpose of pharmaceutical analysis? A. To develop new drugs. B. To ensure the quality of pharmaceutical products. C. To market pharmaceutical products. D. To conduct clinical trials on drugs. The correct answer is B. Pharmaceutical analysis aims to ensure the quality and safety of pharmaceutical products through various analytical techniques. Which of the following is a common analytical technique in pharmaceutical analysis used for quantification based on the interaction of light with a sample? A. Nuclear magnetic resonance spectroscopy. B. Flame atomic absorption spectroscopy. C. O-visible spectroscopy. D. X-ray diffraction. The correct answer is C. O-visible spectroscopy is commonly used in pharmaceutical analysis for quantification by measuring the absorption of light at specific wavelengths. Why is method validation important in pharmaceutical analysis? A. To speed up the drug development process. B. To verify the accuracy and reliability of analytical methods. C. To comply with regulatory requirements. D. To reduce costs in pharmaceutical production. The correct answer is B. Method validation is essential in pharmaceutical analysis to verify that analytical methods are accurate, reliable, and suitable for their intended purpose. Which analytical technique in pharmaceutical analysis involves measuring the size distribution of particles based on the scattering of light? A. Chromatography. B. Mass spectrometry. C. Dynamic light scattering. D. X-ray crystallography. The correct answer is C. Dynamic light scattering is commonly used in pharmaceutical analysis to determine the particle size distribution of samples. What is the main goal of stability testing in pharmaceutical analysis? A. To determine the ideal storage conditions for drugs. B. To assess the long-term stability of pharmaceutical products. C. To speed up the drug registration process. D. To decrease the production costs of drugs. The correct answer is B. Stability testing is conducted in pharmaceutical analysis to assess the long-term stability and shelf life of pharmaceutical products. In the context of pharmaceutical analysis, what does GCM stand for? A. Gas chromatography mass spectrometry. B. Glucose control medical screening. C. General compliance microbiology system. D. Gel column molecular separation. The correct answer is A. GCM stands for gas chromatography mass spectrometry, a powerful analytical technique for separating and identifying compounds in pharmaceutical analysis. Which type of analysis focuses on identifying the presence or absence of specific elements or functional groups in a pharmaceutical sample? A. Quantitative analysis. B. Qualitative analysis. C. Chromatographic analysis. D. Spectroscopic analysis. The correct answer is B. Qualitative analysis in pharmaceutical analysis focuses on identifying the presence or absence of specific elements or functional groups in a sample. What role does a reference standard play in pharmaceutical analysis? A. To compare the results of different analytical techniques. B. To calibrate instruments and establish measurement accuracy. C. To speed up the analysis process. D. To reduce the cost of analysis. The correct answer is B. Reference standards are used to calibrate instruments, establish method accuracy, and ensure the reliability of results in pharmaceutical analysis. Which factor is essential when selecting an analytical technique for pharmaceutical analysis? A. Cost effectiveness of the technique. B. Availability of the equipment. C. Specific requirements of the analysis. D. Speed of the analysis. The correct answer is C. The specific requirements of the analysis, such as the type of sample and the information needed, are critical factors in selecting an appropriate analytical technique for pharmaceutical analysis. What is the main focus of method development in pharmaceutical analysis? A. To increase the sensitivity of analytical instruments. B. To optimize the analysis time. C. To develop reliable and accurate analytical methods. D. To reduce the cost of analysis. The correct answer is C. Method development in pharmaceutical analysis focuses on creating reliable and accurate analytical methods to meet the specific needs of the analysis. What does HPLC stand for in the context of pharmaceutical analysis? A. High pressure liquid chromatography. B. High performance liquid chromatography. C. Hydrophobic liquid chromatography. D. High precision liquid chromatography. The correct answer is B. HPLC stands for high performance liquid chromatography. It's a common analytical technique in pharmaceutical analysis for separating and quantifying components in a sample. Which technique in pharmaceutical analysis involves the separation of components based on differences in their boiling points and vapor pressures? A. Spectroscopy. B. Gas chromatography. C. Mass spectrometry. D. Titration. The correct answer is B. Gas chromatography is the technique in pharmaceutical analysis that involves separating components based on differences in boiling points and vapor pressures. What is the primary function of the O-Vis spectrophotometer in pharmaceutical analysis? A. Determine molecular weight. B. Quantify elemental content. C. Measure light absorption. D. Analyze particle size distribution. The correct answer is C. UV spectrophotometers are commonly used in pharmaceutical analysis to measure the absorption of UV or visible light by a sample. This helps in quantification and identification. Which technique is commonly used in pharmaceutical analysis to identify and quantify elements present in a sample based on their atomic mass and concentration? A. NMR spectroscopy. B. ICPOES. C. Chromatography. D. X-ray crystallography. The correct answer is B. Inductively coupled plasma optical emission spectrometry. ICPOES is commonly used in pharmaceutical analysis to identify and quantify elements based on atomic mass and concentration. What analytical technique in pharmaceutical analysis uses a high-energy beam of electrons to determine the chemical composition of a sample? A. FTIR spectroscopy. B. X-ray diffraction. C. Scanning electron microscopy. D. NMR spectroscopy. The correct answer is C. Scanning electron microscopy is a technique in pharmaceutical analysis that uses a high-energy beam of electrons to determine the chemical composition and structure of a sample. Which technique measures the speed of sound in a medium to determine the physical properties of a sample in pharmaceutical analysis? A. Acoustic spectroscopy. B. Mass spectrometry. C. Nuclear magnetic resonance. D. Atomic absorption spectroscopy. The correct answer is A. Acoustic spectroscopy is an analytical technique used in pharmaceutical analysis to determine physical properties of a sample by measuring the speed of sound in a medium. What does FTIR stand for in the context of pharmaceutical analysis? Functioning transmittance infrared, full transmittance infrared, Fourier transform infrared, frequency transformed infrared. The correct answer is C. FTIR stands for Fourier transform infrared, an analytical technique commonly used in pharmaceutical analysis to study the chemical structure of a drug. Which technique in pharmaceutical analysis involves measuring the heat flow associated with chemical reactions or physical changes in a sample? A. Mass spectrometry. B. Differential scanning calorimetry. C. UV-Vis spectroscopy. D. Titration. The correct answer is B. Differential scanning calorimetry is an analytical technique in pharmaceutical analysis that measures the heat flow associated with chemical reactions or physical changes in a sample. Which of the following is a common application of gas chromatography in pharmaceutical analysis? A. Particle size distribution analysis. B. Determination of molecular weight. C. Identification of functional groups. D. Separation of mixtures based on boiling points. The correct answer is D. Gas chromatography is commonly used in pharmaceutical analysis to separate and quantify mixtures based on their different migration rates in a solvent. Which technique is commonly employed in pharmaceutical analysis to identify and quantify specific functional groups present in drug molecules based on the absorption of infrared light? A. Nuclear magnetic resonance, NMR. B. Ultraviolet visible, UV spectroscopy. C. Mass spectrometry, EMS. D. Fourier transform infrared, FTIR spectroscopy. The correct answer is D. Fourier transform infrared, FTIR spectroscopy is a non-destructive analytical technique often used in pharmaceutical analysis to identify functional groups present in a drug molecule based on the absorption of infrared light. What role do reference standards play in pharmaceutical analysis? A. Setting limits for method validation. B. Ensuring accurate quantification of analytes. C. Identifying unknown compounds. D. Comparing results to known substances. The correct answer is B. Reference standards are used in pharmaceutical analysis to ensure accurate quantification of analytes by comparing the sample response to the reference standard response. Which factor is critical when selecting an analytical technique for pharmaceutical analysis? A. Speed of analysis. B. Cost of equipment. C. Sensitivity and selectivity. D. Sample volume required. The correct answer is C. The sensitivity and selectivity of an analytical technique are critical factors to consider when selecting a method for pharmaceutical analysis. What is the main purpose of stability testing in pharmaceutical analysis? A. To determine the molecular weight of a compound. B. To investigate the speed of sound in a medium. C. To assess the physical properties of a sample. D. To evaluate the long-term stability and shelf life of a pharmaceutical product. The correct answer is D. Stability testing in pharmaceutical analysis is conducted to evaluate the long-term stability and shelf life of a pharmaceutical product under various conditions. In pharmaceutical analysis, what does ICPM stand for? A. Inductively coupled plasma mass spectroscopy. B. Ion chromatography mass spectrometry. C. Infrared chemical profiling mass spectrometry. D. Internal calibration protocol mass spectroscopy. The correct answer is A. ICPM stands for inductively coupled plasma mass spectroscopy, which is commonly used in pharmaceutical analysis to determine the molecular weight of compounds by measuring the separation of ions based on their mass-to-charge ratio. Which type of analysis focuses on identifying the presence or absence of specific elements or functional groups in a pharmaceutical sample? A. Quantitative analysis. B. Qualitative analysis. C. Spectral analysis. D. Chromatographic analysis. The correct answer is B. Qualitative analysis in pharmaceutical analysis focuses on identifying the presence or absence of specific elements or functional groups in a sample without determining the quantity. What is the primary focus of method development in pharmaceutical analysis? A. Quantifying analytes in a sample. B. Identifying unknown compounds. C. Optimizing analytical techniques. D. Ensuring long-term stability of products. The correct answer is C. Method development in pharmaceutical analysis focuses on optimizing analytical techniques to achieve accurate and reliable results for the analysis of pharmaceutical products. What does HPLC stand for in the context of pharmaceutical analysis? High-performance liquid chromatography, high-powered light spectroscopy, hydrogen peroxide level determination, hepatic enzyme profiling. The correct answer is A. HPLC stands for high-performance liquid chromatography. It's a commonly used analytical technique in pharmaceutical analysis for separating and quantifying components based on their interactions with a stationary phase and a mobile phase. Which analytical technique is commonly used in pharmaceutical analysis for the separation and quantification of compounds based on their mass-to-charge ratio? A. Gas chromatography. B. High-performance liquid chromatography. C. Mass spectrometry. D. Thin layer chromatography. The correct answer is C. Mass spectrometry is commonly used in pharmaceutical analysis for its ability to separate and quantify compounds based on their mass-to-charge ratio. What is the term used for the process of confirming the quality and reliability of an analytical method in pharmaceutical analysis? A. Quality assurance. B. quality control C, method validation D, method verification. The correct answer is C. Method validation in pharmaceutical analysis refers to the process of confirming the quality and reliability of an analytical method. Which analytical technique in pharmaceutical analysis uses a high-energy beam of electrons to determine the chemical composition of a sample? A. Nuclear magnetic resonance spectroscopy. B. Electron paramagnetic resonance spectroscopy. C. X-ray diffraction. D. Electron microscopy. The correct answer is D. Electron microscopy is an analytical technique in pharmaceutical analysis that uses a high-energy beam of electrons to determine the chemical composition of a sample. Which factor is crucial when selecting an analytical technique for pharmaceutical analysis? A. Cost effectiveness. B. Speed of analysis. C. Sensitivity and selectivity. D. Availability of equipment. The correct answer is C. The sensitivity and selectivity of an analytical technique are crucial factors to consider when selecting one for pharmaceutical analysis. What is the primary objective of stability testing in pharmaceutical analysis? A. To assess the purity of a sample. B. To determine the molecular weight of a compound. C. To evaluate the chemical and physical stability of a pharmaceutical product. D. To quantify impurities in a sample. The correct answer is C. The primary objective of stability testing in pharmaceutical analysis is to evaluate the chemical and physical stability of a pharmaceutical product over time.